Hi, welcome to Physionic, where we learn the body from the macro to the micro. If that's something you think you'd be interested in, then consider subscribing. Welcome to this examined content, wherein we'll be discussing this particular paper, breaking down figure by figure this paper that looks at how psychedelic drugs, meaning LSD, DOI, DMT, as well as shrooms, and how those impact our brain. So if that's what you're here to find out about, then in this examined content, that's exactly what we're going to find out. So without further ado, let's jump into this examined piece. So let's jump into a bit of the methods. What different styles of investigations or experiments that they use? Well, one, they used a rat model, so obviously using different types of rats. And then they also used an in vitro model, taking primary neurons out of the rats, so out of their brains, and then plating them and then actually doing experiments on the neurons themselves. And they also used a fly model. Now I understand that some people understand why we would use a rat model because it's a mammal, somewhat similar to what we are, but in terms of flies, that doesn't make much sense because they're insects. Well, the reason why is because of a conserved neural circuitry. So some of the circuitry that's found in flies is actually applicable to humans as well. Of course, we are far, far more complex than flies, but because flies are so simple, it's easier to measure in a model like that, because you're not going to have a bunch of divergent pathways or divergent explanations for the results that you're going to get. So if you get results, then you can bring that to a mammalian model like a rat, and then from there, from that animal model to an actual human model, and see if it's a conserved pathway that you see kind of across several different types of species. And in terms of the actual measurements, well, they injected the drugs, the DOI, LSD, and DMT, as well as others, but those are the three that I'm going to be focused on. They also compared it to ketamine, which is an antidepressant, a ever popular antidepressant, to see how these three drugs against ketamine. They don't do a whole lot of measures in that area, but they do a few. And then they also do voltage clamp, where they essentially clamp a particular neuron, and then they're able to tell the actual frequency and amplitude of how that neuron is behaving based off of the drugs that are administered. And then they also did microscopy work, where they did imaging studies, where they were able to fluoresce particular proteins to figure out where those proteins are going, as well as where they're located and how much of those proteins are present based off of the administration of these particular drugs. And finally, they also added some inhibitory drugs to find the mechanisms of how some of these drugs work, but I will go over those as we get into those particular figures, because it's kind of pointless to just throw a bunch of garbage information at you right now, but rest assured we will of course cover it. So with that said, let's jump into the results. All right, let's go into the results. Now, before we actually jump into the results, I think it's important that we get some basic neurology in terms of essentially everything that they're going to look at is looking at neurons. So what is a neuron, and of course kind of the basic structure, the morphology of a neuron. Now there's three distinct parts. There's the dendrite that receives signals, so that's all you need to know. The dendrites receive signals, and that's where we're going to be focused on. The soma, or the cell body, is essentially the integration of the different signals. So you have many dendrites that go off of the soma, so many different inputs of signals. The soma then decides, in a manner of speaking, okay, am I going to continue this signal, or am I not going to continue this signal that I'm receiving from all these dendrites? And then the axon is the actual sending or the propagation of that signal. So if the soma decides to continue the signal, then you get a signal that moves down the axon, and then that axon communicates to the next neuron on the dendrite, obviously, of that next neuron, because it's going to be receiving signal. So reception decision and then sending that signal forward. That's all you need to know, but it's important. So they're going to mention things in the paper like dendrite arborization, which is a fancy term for saying dendrite branching. So the dendrites, the receiving signals, you're getting more of those dendrites as they're kind of branching out. They're becoming more complex. They have a greater ability, presumably, to receive signals from other neurons. So in figures 1A and B, they're essentially just showing those are the 3 drugs they're going to be investigating. And I don't really know why they added this in here for 1B, but they showed an illustration of what will happen. But we're actually going to see images, like real microscopy images of what actually happens with the administration of these drugs. So this was actually pretty worthless information. But whatever, they added their own illustration of what they think or what actually ends up happening. So then 1C is essentially a graph of the 3 different drugs. So if you administer these drugs, and this is in vitro, so on cells, as well as in vivo on the rats themselves. So the administration of these drugs leads to a greater complexity of the dendrite. So you're seeing greater dendrite arborization, greater dendrite branching with all 3 of these drugs compared to control, meaning that the administration of the vehicle is what the drug is, kind of the solution that it's in without the drug itself. So that's why it's called a control. So what's actually happening compared to that control? And well, you see that there's elevations in complexity of those dendrites and kind of the change in morphology of those dendrites with the addition of all 3 drugs. Then for a bunch of figures afterwards, figure D, E, F, so on and so forth, they're essentially just quantifying what is happening to the dendrite's length as well as their width, so the thickness of the dendrite. So are they getting larger or are they getting smaller, or are they staying the same? And compared to vehicle, again, all 3 drugs show that there's an increase in the length. The dendrites are becoming longer as well as thicker with the administration of any of these drugs, which is a really interesting result compared to control. Then for figures 1J and M, they're looking at a fly model here. So they're looking at what is happening to flies at particular stages in their development. So the first instar is considered the first stage, essentially. And I'm certainly no fly expert, but it is a developmental stage that is early on. And here they're measuring just LSD and DOI, and we're seeing greater dendrite complexity and growth with the administration of both of those drugs at the beginning of development, that first instar, but also with the other graph, you're seeing a third instar, which is later on in development. So a little bit more mature fly, you're still seeing the same results. And then they've got actual microscopy images of the neurons, and you can tell that there's greater complexity. You're seeing more branch points off of the neurons that have been given the drugs, the psychedelic drugs, LSD and DOI, as opposed to the vehicle. So really interesting that you're actually seeing some of these changes occurring. For figure 2, you're seeing a lot of characterizations of kind of the morphology. So again, kind of changes that are happening, but from a more zoomed in approach. So they're looking at microscopy images, they're expressing particular proteins, fluorescent proteins, to be able to image the neurons or parts of the neurons themselves. So for example, A and B, you're seeing that they're administering all 3 of these drugs, DOI, LSD, DMT, compared to vehicle. And you're seeing that there are changes in what's called synaptogenesis and spinogenesis. So spinogenesis is the creation of dendritic spine. So it's kind of the beginning of this arborization process. It's the beginning of this branching process. So you're getting some kind of buds almost. Think of like a plant in a way, that you're seeing this kind of initial sprout that's occurring. And the morphology of that sprout is actually very important because you have different stages in which it's extremely immature to more mature. And interestingly enough, the vehicle or the control condition shows more proportion of more mature versions of these sprouts, these dendritic spines, compared to essentially all three of the other drugs. Now, this could be a kinetic issue wherein they're administering the drug and it's not being given enough time to get into that mature state. So that's why you have a greater proportion of these immature sprouts or dendritic spines compared to control. So it could be a kinetic issue, but here you're seeing something that would be presumably in favor of a control, so a non-use of drugs as opposed to the use of any of these three psychedelic drugs. Then for 2I, you're seeing that they're showing again microscopy images, but without necessarily fluorescing the different proteins. This is just a straight light microscopy, meaning that they're just showing some light just to be able to see the actual image. And you're seeing that by comparison to control, you're seeing a lot more of these dendrites kind of branching off of this particular segment of the neuron. And then they quantify some of that in figure 2J, and this is just with DMT, so they didn't look at LSD or DOI for this. And they also included ketamine, which is what they're actually really interested to find out about, because ketamine is a potent antidepressant, and they want to know how these psychedelic drugs stack up against this potent antidepressant. And it seems like DMT may be a little bit less effective than ketamine itself, but both of them are significant compared to control. So you're seeing improvements, or maybe not necessarily improvements, I should say, but greater complexity if that translates to improvements with ketamine and with DMT. So in figure 2, they did a lot of comparisons to ketamine. Well, maybe not a lot of comparisons. They did a little bit of comparison to ketamine, which is again that antidepressant. Now for figure 3, they want to find out how these psychedelic drugs stack up against the endogenous, or the within the body production of neurotogenesis stimulating molecules, meaning the production of more neurons. So the actual creation, the synthesis, the growth of more neurons going on in our brain. And they want to see that using a extremely popular molecule known as BDNF. BDNF is brain-derived neurotrophic factor, and it's widely regarded as a molecule that our body produces that is highly beneficial for brain health. So they're showing different concentrations in the first few figures or first few segments of this figure, where they're seeing increases in BDNF, and they want to know, okay, is that going to have an impact on this neurotogenesis? So looking at BDNF in those first few segments of this particular figure, you're seeing added concentrations of this BDNF and its impact on neuridogenesis, or neurogenesis essentially, where you're getting this increase in neurons. And what you're seeing is that with increased concentrations of BDNF, well, no surprise there, of course, you're going to see increases in these neuronal markers, these neuronal synthetic markers. So you're seeing increases in neuron production, but then does that translate to these drugs? How do these drugs stack up, the psychedelic drugs stack up against BDNF, that endogenous production? And what they find is, by comparing DOI alone, you see roughly the same effect that you do with BDNF. So BDNF and DOI seem to be just about equally effective at promoting this neurotogenesis. But if you combine both of them, you do not see an additive effect. So they seem to be capped at that particular level. So you're not going to see, if you increase BDNF and you take DOI, you're not going to see greater neurogenesis. You're not going to see better improvements in terms of that synaptic growth. Figure 4 is where they're starting to get into the mechanisms. So they know that these drugs have particular impacts, but how do these drugs actually function? How are they going about their cellular signaling to cause this increase in neuronal growth? And they're testing all three of these particular psychedelic drugs. And they hypothesize that it's using a receptor on the cells, on the neuronal cells, that is akin to and is used by BDNF, so what we looked at in the previous figure. And although BDNF can bind this receptor called TRKB, TRKB will then bind to BDNF and then lead to this translation of BDNF's impact or signaling into the cell. So then the cell will suddenly start to communicate to a bunch of other molecules, okay, we need to do something, we need to promote growth. So what they do is they use an inhibitor known as ANA12. ANA12 will inhibit this TRKB receptor so that anything that binds the TRKB receptor will then be nullified. So it will bind, sure, but it can't actually perform its function within the cell. So they want to know if these drugs, by inhibiting TRKB, if they're still going to have an impact on this neuronal growth. And what you find is that with the addition of this inhibitor, it knocks down the effect of two of these psychedelic drugs. DOI and LSD are rendered mute because of this inhibition. So that lends some evidence that they do function through a BDNF style pathway. They use this TRKB receptor, or at least some of the cell signaling within, that is akin to BDNF. But that's not the only story here, because DMT seems to have a blunted effect, but not a complete amelioration of its effects. So it does still seem to have some effect. So that would then imply that DMT has an independent pathway as well as potentially a partially dependent pathway on this TRKB receptor. Okay, so now figure 5 is where they're using another drug to inhibit our psychedelic drugs in whatever capacity they want. So in this particular instance, they're using what's called rapamycin. Rapamycin will act on mTOR. mTOR is the master regulator of growth in cells in general. So of course, you're not going to see much neuronal growth if you knock out the chief molecule, the conductor, that tells everybody, okay, you need to start growing. So this drug will inhibit that conductor, will inhibit mTOR. Now they want to know if any or all three of these psychedelic drugs use mTOR exclusively to create this neuronal growth. And when they add rapamycin, what they find is, again, with two of the drugs, you're seeing an amelioration of their effects. So they no longer lead to any sort of extra growth compared to controls. And those two drugs are DMT and LSD. But again, it's not the end of the story, because DOI seems to still be slightly elevated. So you're still seeing some of that synaptogenesis, you're seeing some of that neuronal growth, that neuronal complexity, that dendritic complexity with DOI independent of mTOR. So mTOR is inhibited, and yet you're still seeing some effect. So it may be partly going through mTOR, but it may also have an independent effect on neuronal growth. Pretty cool. Finally, for figure 6, they're again trying to find out some more of these mechanisms for how these psychedelic drugs are working. So they're using another drug, ketanserin, and that is going to inhibit the serotonin receptor. So serotonin, which is a molecule that you may be familiar with, binds this particular receptor, and then, just like BDNF and TRKB, will lead to a cell signaling cascade, which will tell the cell what to do. Now, when serotonin binds, it binds at that particular site, but if you inhibit that site so that nothing can bind to it, or even if it does bind to it, it somehow doesn't function, then of course you've inhibited that entire pathway, or at least part of that pathway. And they want to find out, so is then, are any of these psychedelic drugs mediated, or do they use serotonin receptors in some way? And it seems that with all three drugs, you see an inhibition of their effect on neuronal growth with the blockage of that 5-HT, that serotonin receptor. So, in conclusion, what can we take away from this? Oh man, there's a lot of information here, and clearly I didn't go over every single figure, but of course the paper is linked for you to investigate every single figure, and I encourage that you do. But what can we take away from what we've gone over? Well, we know that these drugs do have an impact. Psychedelic drugs have an impact on our neurons. They tend to lead to this greater arborization, this greater branching of our dendrites, or of mammalian dendrites in rats, if that translates to humans, which is, as usual, a big if. But if that were to happen, that would be an interesting result. So no doubt these psychedelic drugs, if that's LSD, DOI, or DMT, all of three of them have an impact on our neurons. We also know that these psychedelic drugs have an impact on neurons that is very much akin to BDNF, that endogenous, self-created, brain-derived neurotrophic factor. We also saw a brief comparison to ketamine, which is a potent antidepressant, and with DMT specifically, not tested in LSD or DOI, in what I presented, we saw that DMT has similar effects as ketamine. So whatever ketamine is doing to neurons, a similar system or similar effect is happening with DMT, which is really, really cool and could have some serious implications down the road. Now, another thing to say, and this is something that they said in the introduction of the paper, and I didn't mention this before, that 1/3 of individuals that suffer from depression don't get any benefit from many different types of antidepressants except for ketamine. Ketamine seems to help that 1/3 of the population, which is a massive amount of individuals. So if now they're showing a lot of similarities between these psychedelic drugs and ketamine, maybe there's a potential out there that these psychedelic drugs could be used in place of ketamine and have a positive impact against depression. Again, if this were to translate to humans and if these effects are actually considered positive, because all we're saying here is that we're seeing changes. We're seeing changes in morphology. We're seeing changes in terms of the complexity of our neurons. And a lot of this is associated with some of these antidepressants that we've been using for a number of years now. So if that's causative, we can't 100% say. I just want to be really clear about that, because I don't want people walking away from this thinking psychedelic drugs are immediately, oh, absolutely, they're exactly the same as antidepressants. There's some evidence here for that, but we can't conclusively say that without more studies. Another thing I want to add in here is something that they mentioned in the introduction as well, that some of these drugs have a very short half-life. They don't stick around in our system for very long, some as low as 15 minutes, and yet the effects stand out for hours afterwards. So they may quick enter our system, then come out, and still the effects continue. There's a protective, potentially protective effect that continues going forward for many hours. And then the dosages, especially for LSD, is incredibly small compared to really any of the other psychedelic drugs as well as antidepressants. So you're using much smaller doses and you're still seeing equal, if not better, results from that psychedelic drug. So finally, can we say psychedelic drugs improve depression? No, we can't. Even though they do mention that with depressive individuals you see atrophy of neurons, so you're seeing the degeneration of neurons, and here we're seeing evidence that there's a synthesis, or the opposite, a benefit to the neurons in terms of their complexity. We're seeing those changes, but we can't 100% translate that immediately to betterment in outcomes, in terms of emotional outcomes, in terms of stability of our mentality and things of that nature. But it's definitely encouraging to see that some of these changes are certainly in line with a lot of antidepressants. So hopefully you found this informative. I really do hope that you thought that this was as interesting as I did. I certainly learned a massive amount of information based off this paper. And with that said, I hope I have the absolute pleasure of speaking with you in the next one. Have a good one, guys. See ya.